Welcome to the first PowerPoint for Unit 3 Biology. So Unit 3 is how do cells maintain life, and our first topic is plasma membranes. So here we have the key knowledge checklist. This comes directly from the study design. You should have a copy of this printed out, and I recommend gluing it on the first page before you start taking notes for this particular topic. As you feel as though you have mastered any one of these three, make sure you tick it off that list, and hopefully that should make revision a little bit easier for you later. Alright, so starting with the structure of the plasma membrane, the basis for the plasma membrane is something called a phospholipid, and the phospholipid has a hydrophilic head, which means it's attracted to water, and it contains phosphorus in this particular section, and it has a hydrophobic tail. So the hydrophilic section is the blue bit, this top part up here. So this is the bit that likes water and has the phosphorus, and then down the bottom here we have the hydrophilic tails. So they're the hydrophilic tails, and they don't like water, but they contain lipids, so they contain a fat, which this structure is very important for enabling things to come into and out of cells. So the hydrophilic head attracts water, the hydrophobic tails repel water. Very important to remember that. So we use the term fluid mosaic model when we are talking about the structure of the plasma membrane, and what we are talking about here is that the phospholipids allow for flexibility of the membrane. So as you can see in the picture to the side, it's not a solid line like we would be drawing in class. It actually moves around and has lots of things stuck into it. So at the top of the picture we have the external cell membrane, the cell contents, and then the internal cell contents at the bottom. And you can see that there are proteins and things floating in the membrane, and none of these things really sit still. And it's not just phospholipids that we were just talking about before, but it's a whole range of things scattered throughout this membrane, giving it this mosaic-like pattern. So it's the glycoproteins and other protein molecules, carrier protein channels that are helping molecules to pass into or out of cells that help give it this mosaic-like appearance. So here we have the parts to the plasma membrane. It's really important that you understand what all the different parts are, because we are going to be looking at modes of transport. So we start with the glycoprotein at the top left here, which is a protein that has a carbohydrate head. So the carbohydrate head is this section just here, and the protein is the big glob that it has at the bottom. We have a glycolipid with the carbohydrate attached, again just here to the right. Down the bottom, from the bottom left, we have a peripheral membrane protein. What that is it's towards the outside, and it's just a protein that sits in the membrane. We have integral membrane proteins. We have cytoskeleton filaments. This help to give structure to our cells. We have cholesterol, which help with the fluidity and movement of the plasma membrane, and you can see a few of these scattered throughout the cell. And then we have protein channels here that are allowing different molecules to enter and leave. And then we have the phospholipid bilayer, bi meaning two. So as you can see, there's the two pairs of phospholipids with their heads facing away from each other and their tails facing towards each other. So the part that likes water is always on the outside, and the part that dislikes water is always in the middle. If I was you, I would be recommending that you make sure that you're aware of all the different protein structures and all the different parts of the plasma membrane and what they do. This is really important, because if you're being asked a question as to how a lack of something enables transport or disables transport or influences the transport of things into and out of cells, you really need to make sure you know these things. So a carrier protein as opposed to just a channel protein. For example, a carrier protein, it needs to bind to it in order to move from one side to the other, where a channel protein is just a pore or a hole that allows things to pass through it. So how do these substances actually make their way across the membrane? I've been saying this for 3 or 4 slides now, that our big focus is how things are able to get across this plasma membrane, but how do they actually do it? So we have a few things that influence this movement. We have whether the substance itself is hydrophilic, meaning that it likes water, that it can dissolve in water or appear to be wet by water, and we're looking at things that are polar. Polar means that they have different charges in different areas. So at the top here we have a negative and then we have positive at the bottom, and this is water. So water is a polar molecule. Then we have hydrophobic substances. These things are termed to be ones that hate water. So they repel water. Think of your oils. So if we're mixing oil with water, like we have in this picture, then we're looking at those oils clumping together because they don't dissolve and they don't like water. So these are termed non-polar. So our polar things like water and they dissolve in it. Hydrophobic is non-polar, they dislike water and they don't dissolve in it. Now, whether something is hydrophilic or hydrophobic is quite important because it influences the transport options available to that material. Another thing that influences how things are able to cross the membrane is the size of them. So if we look at something that's quite large, like what we have on the left-hand side over here, versus some things that are quite small, so like oxygen or carbon dioxide, the smaller that something is, the easier that it is for it to pass across the membrane. The bigger that something is, the more difficult that this task then becomes. If something is smaller, then it can easily try and just pass through some of those pores that we were just talking about, those protein channels, or even just straight across the plasma membrane, depending on whether they're polar or non-polar. If something is larger, then we're looking at maybe endocytosis and having to engulf whatever that product is to get it inside. So think of something like a protein or a large carbohydrate like glucose. Being able to get those things in can take a lot more effort from the cell. So that's where we need those carrier proteins, those assisting proteins. We need more facilitated and helped diffusion for these molecules to get across because they're just too big. The other thing that influences how these things move is concentration gradient. So on the left-hand side here, you'll notice that we don't really have too many of these blue dots, and we've got a whole heap on the right-hand side. And if you look at what I've circled, there's a lot less blue dots on the left compared to the right. So we would say that the left-hand side is at a low concentration and the right-hand side is at a high concentration. Why this is important is because things want to move from a region of a high concentration to a region of a low concentration. They're always trying to reach an equilibrium point, like a balanced point where everything is equal and fair and even. This doesn't mean that things don't automatically stop moving once they've reached this equilibrium point. They will keep moving. These molecules move all the time. It's just that they've reached a point where there's no net movement, so no overall movement. So things will move preferably from a high concentration to a low concentration. And this concentration is quite important. It influences chemical reactions and those types of things in the body. So diffusion is our first mode of transport that we're going to be looking at, and it can also be known as simple or passive diffusion. Really important that you know the definition of this one. It's the net or overall movement of particles from a region of high concentration to a region of lower concentration, like we were just talking about before. And as you can see in the two pictures, there's less dots at the top compared to the bottom, so they're going to be moving from the bottom to the top. It follows the concentration gradient, so it always goes from a high to a low concentration, and it slows down. So the process of these molecules moving, slows down as they start to reach that equilibrium or balanced or equal point where everything's the same. And it's a passive process, meaning that it doesn't need any energy at all. So no ATP is used in diffusion. So if we look at this one again, there are a few examples of things that can pass through the plasma membrane by diffusion. It's carbon dioxide and oxygen, the two that we were just talking about before, and that concentration needs to be maintained. So if you think about carbon dioxide, it's a waste product in our body. We don't really want it there, but we're always producing it. So how are we able to get rid of it? So what we're looking at is shifting that concentration gradient. So if I have a high concentration of carbon dioxide in my blood, that blood then goes to my lungs where there is a lower concentration of carbon dioxide, so that the carbon dioxide moves from my blood into my lungs and then I exhale it, which then, because that blood moves around, that is then at a lower concentration, enabling the carbon dioxide to move out of my cells and into the blood for this process to then continue. So it's important that these things keep moving, and that concentration gradient has to be maintained so that these molecules can continually be moved, whether it's a nutrient or something that we need. So we need oxygen, so the same thing has to happen there. We need a lower amount of oxygen in our blood to allow it to diffuse from our lungs into our bloodstream. So this concentration gradient is very important for diffusion. So our next mode of transport is facilitated diffusion, and this one uses transport proteins. So those proteins that we found in the plasma membrane, where we were talking about either carrier or channel proteins, and they help things to move into or out of cells, and generally they're a little bit larger. So think glucose, amino acids, nucleotides, or things that are charged, so ions like our salts. Our carrier proteins, when we're talking about these ones, it's the diffusing molecule that then binds to the carrier protein and is then carried to the other side. These particles can then move with the concentration gradient. So they can't go against the concentration gradient, same as diffusion, goes with the concentration gradient. Our channel proteins form those pores or holes that we were talking about earlier in the plasma membrane, and then those pores fill with water, so that anything that's hydrophilic, anything that likes to dissolve in water, is then able to pass across the plasma membrane quite easily. So what we're looking at here is some examples of those. So we've got the protein channel on the left here. So that's just a simple hole, meaning that anything that's dissolved is able to pass straight through nice and easily. And then we've got the channel protein. So the molecule will then enter, it would then be helped to exit the other side. And again, with the concentration gradient. So there's more stuff on the outside and it's all moving in this example. Not always the case though. It can go the opposite direction, depends on the substance and the concentration gradient. Osmosis, this is the net or overall movement of water molecules across a differentially permeable membrane. So they're permeable to some things but not all, from a dilute solution to a concentrated solution. So in a dilute solution, that means that there is more water compared to what's dissolved in it when you look at it next to a concentrated solution. So if you think about cordial, in a dilute cordial, you don't have very much. If it's concentrated, you have a lot more cordial compared to water. So what's happening here is that the water moves from somewhere where there is a lot more water to somewhere where there is less water compared to what's dissolved into it. So if we're looking at something that has more water, less solute, we refer to that as hypotonic, so less things dissolved, less solutes. And a hypertonic is one that has more solutes or less water. So make sure that you're aware of the differences between the two, hypotonic and hypertonic. It's really important that you remember that water is essential for all life. It allows biochemical processes to take place. It transports materials, like we were just saying before, through those channel pores and all those types of things. It helps to keep our cells shape. So think about our red blood cells, and I spoke about this last year, I'm pretty sure, that they can pop if there's too much water, and that they can shrivel up and not really be able to perform their functions if there's not enough water. And it forms the fluid that bathes tissues. So what that means is that it sits around our tissues as well. So it helps keep them safe and protected from each other, so that there's no real friction going on. And water is described as a universal solvent because it is polar. And because it's polar, that means that anything else that's polar can dissolve into it. So think table salt, NaCl. That one's polar because it's able to dissolve into water, and it has charges. So the charges that water has enables it to pull other things apart and kind of break them down based on the charges that it has. So key terms, please make sure you know all of these: solute, solvent, solution, isotonic, hypertonic, and hypertonic. You need to know the differences between all of those terms. Have a really good read, and please make sure you write them down. Put them onto your flashcards if you need to. Okay, so let's have a look at some of that terminology in action. So we have hypertonic. In a hypertonic, there is more things generally outside the cell than inside the cell. So what that means is that water is going to be leaving the cell. So if there's more stuff on the outside compared to what's on the inside, then water is going to move out to try and get that balance, meaning that particularly in a plant cell, its cell membrane will start to pull away from the cell wall, and it can become what we term as being like a flaccid cell. It's quite soft. Isotonic is when everything is equal and they are all happy, and water just continues to move in and out of these cells at the same rate, meaning that the cells look quite happy. And the last one is hypotonic. So in a hypotonic solution, what that means is that there's more stuff inside the cell than there is outside the cell. So water is then going to be moving into the cell. If we're looking at an animal cell, we don't have a plasma wall, a cell wall. So that means that there's nothing to really prevent these cells from continuing to expand. Water keeps flooding in if it's in a hypertonic solution, and these cells can burst and pop and release their contents. And that can be quite bad if you don't have enough cells to be replacing them. So red blood cells, for example, are made in your bone marrow. These cells are made fresh so that they're able to perform their function. But if it's not able to be sustained, so say all these cells keep popping, it's quite bad for you. You won't be able to carry your oxygen around. In a plant cell, they're a lot luckier. They have a cell wall that prevents them from expanding too much. So when animal cells swell, like we were just saying before, they can swell much more than plant cells because of that lack of cell wall. So if the volume continues to increase, then they can't go any further. There's only a certain amount of expansion that they can have, and then they're going to rupture or burst, or in science terms, lyse. And some unicellular organisms have a constant uptake of water due to the environment that they live in. So think about any bacteria or anything that lives in freshwater environments. They're going to be taking in a lot of water from what's around them. So they have to have vacuoles that are readily able to pump out this extra fluid, and they're termed contractile vacuoles. So they kind of like squeeze out and push out all this extra water that these cells are taking in. So a cell that's in a freshwater environment will take in more water, store it in a vacuole so that it can then remove it later on. So in a plant cell, it's these vacuoles that help keep the plasma wall against the plasma membrane, against the plasma wall, and the water molecules diffuse in or out exactly the same as in an animal cell by the process of osmosis. When the vacuole swells, it pushes that plasma membrane against the wall, and that's what we call the cell to be turgid. So it's quite stiff. So this cell here on the left is quite stiff. Its vacuole is very full. The membrane's all up against the cell wall. The turgor pressure is then exerted to keep these cells firm. So when it is turgid, it is under high turgor pressure. And then the water can also come out of these cells. Remember, things go both ways, not just the one. And then the plasma membrane can pull away from the cell wall, and this is what we call plasmolysis. So then the cell tends to shrivel up a little bit, becomes quite weak. It can't hold its structure. So think about plants that don't have enough water. So out in my garden, I'm not a very good gardener. I have some flowers and things that I sometimes forget to water, and then they start to shrivel because their cells don't have the same strength that they had when they were full of water. All right, active transport. So this is when molecules move against the concentration gradient. Very important difference here. Active, they're going against the concentration gradient, and they need energy. So they need ATP to be able to do this process, and that energy comes from mitochondria. You might remember cellular respiration. We are going to be covering it again, which is how cells get their ATP from glucose. We're looking at carrier proteins here. So we talked about carrier proteins in facilitated diffusion. Now don't get the two confused. Facilitated diffusion does not require energy, but still uses carrier proteins. Active transport uses carrier proteins and does need ATP. So it's a different carrier protein, not the same one for each thing. So that every molecule has its own little carrier to help it across, basically. So when do we need this active transport? Seems like a bit of a waste. When we are digesting our food. So you don't eat all the time, but you constantly need to be taking in sugar. So when you eat, you have a high amount of sugar and glucose and all those types of things in your stomach, and you need to get that into your body so you're able to do something with it. So that glucose molecule is then actively transported into cells when you're digesting things, when you're reabsorbing anything from the kidneys. So the kidneys are a filtering system. Anything that we need back in our body goes through active transport. The contraction of our muscles. So our muscles contract and our muscles relax. When they are contracting, they require energy to do that process. And when we move impulses, so nerve impulses along those nerve cells. So the use of the sodium potassium pump in our nerve cells, which is what this picture here is to the right, requires energy. And it's a very important part to signal transmission of a nerve signal, which we will again be covering a bit later on. Alright, now last forms of transport are endo and exocytosis. So endocytosis is when things are entering into the cell, and exocytosis is when things are leaving the cell. So endocytosis, we're looking at a couple of examples here. We have penocytosis, so the movement of liquid droplets. Phagocytosis is the movement of solid materials. And then there is receptor-mediated one, which is our final example. So these are for things that are very, very large. So proteins, so like milk proteins, hormones, mucus, digestive enzymes, toxins, growth regulators, all of those things are really big. And this is an active process. If it's active, that means it needs energy. So phagocytosis is the first example that we have here. And we can see that this solid particle gets wrapped up by the plasma membrane to then put it into a vacuole. And penocytosis is almost exactly the same, except it's a fluid instead of it being a solid. And this cell-mediated one, you can see that this little star here has triggered a receptor on the membrane, and then it's been engulfed, so the membrane's kind of pulled in and put them inside. So it's a coated vesicle. It has those protein channels and those receptors and everything in it. So those receptors are triggered by, in this case it's a star, it would be something else inside of a real cell, and then the membrane engulfs it to bring it inside of the cell. Exocytosis is the opposite, it is movement of substances out of the cell instead of things coming into the cell. This page here is just a summary. I find it to be quite good and nice and condensed because it can be quite easy to get them all confused and mixed up. So make sure you go over these and you understand the differences between them all. And again, another summary table. When you're doing your summary notes, it's up to you how you present that information, and you need to find the one that is best for you and the one that helps you remember it the best. All right, moving on, we are also going to be looking at cell organelles because our plasma membrane is just one of those cell organelles. So we have an animal cell, a plant cell, and a prokaryotic cell up here in the top right. So our nucleus, this is a membrane-bound organelle, so it is only found in eukaryotic cells. Remember, the big difference between prokaryotic and eukaryotic cells is that prokaryotic do not have membrane-bound organelles. This includes a nucleus. So in the nucleus is DNA, and DNA in eukaryotic cells is in chromosomes. In prokaryotic cells, it was the circular plasmid structure. The nuclear membrane has pores in it, so it has these little holes that you can see, and this allows substances and proteins to move into or out of the nucleus. So our mRNA is able to come out to be able to be converted into a protein. And the outer membrane of the nucleus sticks to the endoplasmic reticulum so that it's then able to travel throughout the cell. So if you think about your rough ER, it has a ribosome attached. So my copy of my RNA will come out through the pores into the endoplasmic reticulum where there's a rough ER, to rough ER where there's a ribosome, where it then makes a protein. The nucleolus is within the nucleus, and that's that site where it makes that rRNA. So ribosomal RNA to make ribosomes. We are going to be talking about types of RNA and things a bit later as well. So ribosomal and mRNA, which is mitochondrial RNA, are synthesized within the nucleus via the pore, and then it goes from there via the pores out into the cytoplasm. The mitochondria, this is the site of cellular respiration, so where our ATP is produced from ADP, so adenosine diphosphate with that extra third phosphate attached becomes adenosine triphosphate. ATP is what we are able to use to carry out our cellular functions, and they have an outer membrane and a highly folded inner membrane. And mitochondria, we're going to be looking at cellular respiration, and different stages of cellular respiration occur at different locations in the mitochondria. So it's a good idea to understand this general structure because it will help you when we get to cellular respiration. Super important, mitochondria are not found in prokaryotic cells because they are membrane bound. They have a membrane around them. Our next one is a ribosome. This is where we make proteins. They are very, very small and they do not have a membrane, which means they can be found in prokaryotic cells, and they can be free floating or attached to the endoplasmic reticulum, making our rough ER. There are two subunits composed of rRNA and proteins, and cells with high rates of protein synthesis can have a lot of ribosomes. So in our liver, where we tend to break down a lot of protein, create a lot of protein to break things down, my liver cells will have quite a lot of ribosomes. Our next one is the endoplasmic reticulum, which I've been mentioning a few times now. So we have the rough ER and the smooth ER. The big difference between the two is whether they have ribosomes attached or not. Rough ER does have ribosomes, which means it can make protein. Smooth ER does not have ribosomes attached. It's more tubular in shape, as you can see here, and is the site of phospholipid and cholesterol synthesis, and some carbohydrates are going to be made there. Carbohydrates are our CHO groups, and it's also where we store calcium ions. The next one is the Golgi apparatus, also known as Golgi body or Golgi complex, depending on which book you tend to read. So here at the Golgi apparatus is where we are packaging those proteins that we just made from our ribosomes, and we're also looking at packaging lipid molecules. So it's a flattened, kind of like a stack of pancakes being stacked on top of each other, and they kind of resemble little tubes. What happens in this packaging is that they add carbohydrates to the proteins and the lipids, and then they form glycoproteins or glycolipids. And then these molecules are packaged up into vesicles. So we discussed vesicles before and their ability to contain water. So these vesicles can also contain other substances. We looked at endocytosis and how they were stored inside the membrane. So these vesicles are then able to store those products, and then they are then able to attach to parts of the plasma membrane so that they can be released by exocytosis, so removing things, so them leaving the cell, these products leaving the cell. So all eukaryotic cells contain a Golgi apparatus, and the more numerous in cells that have that need to secrete things, so think about your pancreas, which makes a lot of pancreatic enzymes and things like that, they're going to have a high number of Golgi apparatus so that they're able to package and deliver what they're producing. Chloroplast. This one's also not found in prokaryotic cells because it has a membrane. There are three key parts that you need to be aware of. There is the thylakoid membrane, the grana, and the stroma. You need to know the differences between these. Don't forget that your chloroplast is only found in plant cells, and it traps sunlight energy so that photosynthesis can occur here. We are going to discuss photosynthesis again very soon, and you are going to be learning about stages of photosynthesis. So similar to cellular respiration, I strongly recommend that you know the chloroplast back to front. So your thylakoid membrane is the inner membrane that forms a system of stacks. So you can see them all stacked on top of each other just here. And then grana is the stack of the thylakoids. So here we have a thylakoid, and then we have a stack of them which makes a grana. And the stroma is the fluid space between them, so this fluid bit in here. And each of them has a different role in photosynthesis. All right, so we need to get these things out of our cells. So we've made our protein from a ribosome, and its function isn't inside this cell, it's in a different cell. So DNA gets converted to RNA. It's going to be a very simple recovering of this. We're going to cover it again later. So don't stress if it makes no sense now. You just need to think of it in terms of the transport of that protein product out of the cell. So DNA becomes RNA inside the nucleus, but we need to get that product to its target cell. So RNA is going to leave the nucleus into the rough ER, so into your endoplasmic reticulum. The ribosome on the rough ER is going to convert your RNA into a protein. It is then going to be packaged into a vacuole by the Golgi apparatus. and then that little vacuole is able to attach to the plasma membrane for it to be expelled from the cell so it can do its function elsewhere. So we start at the bottom here. My product becomes RNA. My rough ER over here is going to convert it into a protein, which then gets packaged and goes to my Golgi apparatus here, which then package it up into a vesicle with some extra information, so it adds those carbohydrates we were talking about so that they're aware of their destination for it to then leave the cell. And what we have on the right hand side here is this cell's actually receiving protein products from another cell as well. So endocytosis, these things are then entering, and then they're going to things like the Golgi apparatus so that they're told what function and where to go inside this particular cell as well. And that is our first PowerPoint for biology.